Okay, so hi everyone, and welcome to the 13th TIA Centre seminar of the academic year. So my name's Adam Shepherd, and I'm also joined by my co-host here, Jachi, and we're researchers in the TIA Centre. And for those that are new to our seminars, we invite researchers and leaders in the area of computational pathology and associated areas to discuss their work. And I'm really pleased today to introduce Dr. Sahar Nasser. Sahar is a postdoctoral researcher at Emory University's Madabushi Lab, working at the intersection of computational pathology, agentic AI, and translational oncology. She is the creator of SAGE, an agentic framework for biologically grounded and clinically translatable biomarker discovery, with a focus on interpretable and trustworthy AI for cancer care. She's a recipient of the Qualcomm Innovation Fellowship, and her work has been published in leading AI and medical imaging venues. And the title of the talk today is SAGE, Agentic Framework for Interpretable and Clinically Translatable Computational Pathology Biomarker Discovery. So, Sahar, thank you very much for joining us. And with that, I'll pass over to you.
Hi, Adam. Thank you so much for having me. It's my pleasure to present our work at the TIA Center. And let's start. So I'll share my screen. I hope it's visible.
Yeah, we can see that okay.
Okay, great. Just, okay. So I'll introduce our work, SAGE, Agentic AI for Biomarker Discovery in Computational Pathology. I am Dr. Sahar Nasser, a postdoc at Madabushi's lab in Emory University. So first of all, I know that many people who are joining this call might be pathologists and they might not be aware about agentic AI. So I thought I'll start my talk by introducing what is agentic AI. So an agent is an LLM model which can receive a task and think about it, plan steps, and take actions, and finally come up with decisions to resolve the task at hand and deliver the results to the user. So it's a bit different than the traditional AI or LLM, where the user asks a question and the LLM deliver an answer, and that's it. But for agentic AI, we have the core is an LLM model which receives the task, divided into subtasks, then plan the steps to achieve the final goal of this task. And for that, it might need to use external tools to take actions and deliver the final results. So, what makes AI agentic? The first step, the AI should be able to reason about the specific task, to take specific action, and then taking the actions by calling external tools, which are real-world tools. And remember the experience throughout the process so that next time it can use this experience to resolve the future tasks. For example, if we are talking about a coding agent, so the coding agent, the first time you give it difficult code, it might take around 6 rounds, for example, to come up with the final answer. But next time, if you give it similar coding task, it should be taking 1 or 2 rounds to come up with the right answer because it has learned from the previous experience how to solve such a task. And this is what we call adaptation, where the LLM model adjusts his experience based on the experiences he went through solving different tasks. So to summarize, the agent is an LLM model which receives a request from the user, plan an action by dividing the task into subtasks, and use external tools, which are real-world tools, to perform these tasks and come up with the final answer and give it to the user. It will observe as well the results and the feedback from the user, so it will learn from the experience. So how do, when we talk about agentic AI system, we are talking about different agents which collaborate with each other. So how's the collaboration happening? So the agents can be a single agent where you give it a task, it think about it, and call external tools to implement it, then come up with the output. And the task might be a bit complicated. So it consists of subtasks. So we have orchestration subsystem and different agents. These are specialized agents. Each one has its own task. So in this example, we have 3 agents, A, B, and C. So when the agentic system receives a task, it divided into the subtask, and the orchestrator, which is like a boss of the system, is an LLM agent which knows the specialty of each of those agents and send that specific task to a specific agent, and finally understand and gather the output from all of these agents to deliver the final results to the user. And we'll see that in Sage when we talk about the orchestration subsystem of tools. Then we have parallel agents. So in this task, those are fixed agents. We have fixed tasks all the time, but we divide it into 2 agents so we can speed up the process. And then we merge the outputs from these agents to deliver the final results. We have the sequential pipeline where the output, where the work of a specific agent depends on the output from the previous agents. So for example, in Sage, we'll see that the senior scientist will depend on the input from a scientist to perform its task. So it's a sequential pipeline. Then we have the critic or debate agents, where we have multiple agents and mostly like 2. They challenge each other on a specific task. And after some rounds of discussing with each other and challenging each other, they come up with the final answer, which they agree both on it. And finally, we have a human in the loop. So for the very sensitive applications like the medical application, we'll have a human in the loop where the human receives the output of the agent, approve it or disapprove it or give a feedback, and then proceed to the next agents. And we'll see that as well in Sage when we talk about human in the loop. So this is about the agentic AI. So is there any question about the agents before I move on?
I don't think we've got any questions at this stage. Thank you.
Okay, so we'll talk now about a biomarker. So maybe some of you might be AI engineers. They might not know much about the prognostic and predictive biomarkers in computational pathology. So I'll give a short introduction about this. So a prognostic biomarker is a set of features which we extracted from a histopathology image, and these features interact with each other to form a signature. And this signature, we call it a prognostic biomarker if it can stratify the patients into low risk and high risk groups. For example, for recurrence, this biomarker can tell you whether the patient has high probability to get recurrence or low probability to have recurrence. Another type of biomarkers is the predictive biomarker. Similarly, it's like set of features we extracted from histopathology images. We combine those features together, they interact with each other to form a signature, which we call it a predictive signature if it can identify the responders to specific treatment versus non-responders. For example, in bladder cancer, which is the focus of Sage paper, we can identify the patient who responds to immunotherapy and will not respond to the immunotherapy. So for computational pathology, we start with the histopathology image, which is biopsy, digitized, and then we get the histopathology image. And we usually have a researcher in the lab or a group of researchers who think about a hypothesis for what it can be a prognostic or predictive biomarker. And this hypothesis will depend on the experience of this researcher or this group of researchers and the papers they read, basically. Then from the hypothesis, they move to the next stage, which is the coding. So they need to transfer these hypotheses into codes. For example, if we are saying that mitotic index is prognostic in bladder cancer, so for that, I need a code to take the whole slide images, divide it into batches, process it, and then have another code for detecting the mitosis, another code for detecting nuclei. Then we can compute the index and see whether it's prognostic or not. So we have then the stage of the feature extraction, then we have the data analysis. And based on the results, this result will prove whether the biomarker is prognostic or not. So in our example, the mitotic index might or might not be a prognostic. Then the results will go to the researcher. He will analyze them and might refine his hypothesis based on these results. And this whole process will go on and on until we reach into the biomarker, which can be prognostic or predictive, or one biomarker can be even both. So this whole process takes lots of effort, lots of time, many researchers in the loop with different specialties. So can we replace all of these process with a single pipeline end-to-end from the hypothesis generation till the validation of the biomarker? So for that, we present SAGE, which is an agentic pipeline for biomarker discovery and hypothesis generation. So if there is no question on this, I'll talk about SAGE. SAGE is a structured agentic AI system for hypothesis generation and evaluation. So the main problem in biomarker discovery that it depends on the knowledge of the people. For example, the handcrafted features need somebody who is experienced in the domain, like the urologist or the pathologist, etc. And there are other type of the biomarkers, like the ones which depend on the feature vectors from foundation model. The problem with these signatures, though it might be significant and robust, the problem with these, they are not explainable. So the pathologist might not trust them. So with SAGE, we need to automate the process to make it faster and provide a knowledge enough to find novel biomarkers. So SAGE works through 3 different stages. The first one is the knowledge graph construction. Then we move to the hypothesis generation and refinement. And the final stage is the validation. So this is an overview of the agentic AI system, which we call SAGE. The user prompt triggers the system. The prompt can be any question from the user, the pathologist, or the researcher about an interesting question they are trying to answer, or specific biomarker they are trying to discover. For example, if the researcher wants to discover a new biomarker which combines features from histopathology domain as well as genes to come up with the prognostic biomarker of bladder cancer, they can ask the system this question. The first stage of the system is the path generation agent, which extracts specific path from the big knowledge graph, and we'll talk about this in details in the next slide. Then this path, it goes to the ontologist, which analyze each node and each connection on this path and give it definition, and send these definitions to the scientist. The role of the scientist agent is to connect these different definitions and the connection between them and come up with a simple hypothesis. Then the simple hypothesis go from the scientist to the hypothesis expansion agent. So this agent take the output from the scientist, which is the simple hypothesis, and try to ground it in the biology. So to find the references for each relationship or each claim in the given hypothesis. After that, it goes to the human. So the human in the loop can either approve the hypothesis based on his experience, or reject the hypothesis, or ask for refinement, so it goes back to the ontologist, and the scientist, then the research scientist. So this continues, but let's assume that the human approves the hypothesis, then it goes to the novelty critic. So the task of this agent is to decide whether the hypothesis is novel enough or not, because a hypothesis might be visible and the human might like it, but it might not be novel. This hypothesis might be already available in the literature. So this is the task of the novelty agent. And if the hypothesis is not novel enough, it goes back to the scientist to refine its hypothesis, and then we go back to the senior research scientist and the human and the novelty critic until we reach the novel hypothesis. If the hypothesis is novel enough, it goes to the feasibility agent, which check the hypothesis and check if we have available data set, whether it's in our repository or in the online, to validate the hypothesis. And after the feasibility agent, it goes to the coding task agents, which runs the code, and it has access as well to the external tools, which it can just prompt and run those tools on the images or the data set. And after it finishes the coding step, we get the results. So there is another agent to analyze the results and check whether the results are whether the hypothesis is valid or not. So for example, in prognostic biomarker, we have the survival curves. So the agents looks into the p-value, the hazard ratio, and all other things to make sure it's a novel hypothesis or not. Then we go to the summary agent, where we summarize the output from all the agents and all the process. So is there any questions so far? Okay. So
I don't
then
think so. Yeah, thank you.
Okay. So we'll move to the knowledge collection agents. So the first step to design a robust model is to provide it with enough knowledge to come up with novel hypotheses. So the knowledge collection agent is the first agent we have. So we start by asking the expert in the domain to give us keywords in this domain. For example, for bladder cancer, we have a urologist in our lab who is a postdoc, Dr. Nawato, who give us list of the most important keywords in the bladder cancer. So after getting these keywords, we use ChatGPT, Mesh, and Semantic Scholar to expand these keywords. So Mesh is like a database which has all the terms in medicine. So it was very useful to expand the set of keywords which we have. And after expanding the keywords, so we move from 308 given by the urologist to 1,276 keywords. Then from these keywords, we go to the next stage, which is the literature crawling. So in this stage, we first filter these keywords because some of them might be like repeated, or they might not be that related to our domain. So we filter them based on different criteria about how similar these keywords to the domain of interest. As well as we provide a set of best journals in the domain and conferences with their impact factor. So the paper, the keywords which coming from those resources will have higher weight than the keywords which comes from the low impact journal. Then after having the final set of keywords, we end up with 748 keywords, and we use these keywords to download the related papers. So we end up with having papers which can be either digitized PDF, so it's very easy to process and divide the text into chunks, or it can be scanned PDF for old papers and we process it as images. And we have as well the figures and the tables in the paper, so it needs another package for processing them. So while processing them, we divide the text into chunks, and these chunks are overlapped. So it's around 1,500 tokens each, and we select overlapped chunks so that we keep the context from the text. And finally, we get the chunks from the paper. So for the bladder cancer, we downloaded around 137,000 papers. And after all the filtering steps, we end up with the 1,600 papers. So now after having the chunks from those texts, we move into the next step, which is building the knowledge graph. So from the chunks, we generate triples. So each triples, it consists of the head node and the tail node and the connection between them. So for example, if we are talking about a node which can be a gene, like FABP5 here, so this gene might have a link to the tertiary lymphoid structure aggregate, and the relationship between them is like associated with. Or for example, this same gene, it might have a relationship with the overall survival in bladder cancer, and the relationship that the gene predicts the overall survival. And you can see for each of these triples, we have confidence for the connection between them. So if the connection between them is greater, has a confidence greater than 0.5, which is a threshold we selected, we keep this triples in our knowledge graph. Then after this step, we have the second step, which is the blacklist filtering. So in this step, we remove all the small clusters in the graph which are not related to the graph, or not connected with other parts of the graph, which might be noise, or like some concepts which are not very much connected to the topic of the paper. So all of this process have been done on a single paper so far. So we generated a local graph from one single paper or textbook. Then we do the same for all the 1,600 papers we have, and we end up with many knowledge graphs, local ones. Then we merge them together to have the final knowledge graph. So in the final knowledge graph, as you can see, we have different nodes, and we give the nodes types. Like, for example, some of them might be genes, some of them might be disease, some of them might be like pathway, etc. And if you take this small region and zoom it in, you can see how these concepts are connected with each other. So our final knowledge graph consists of 41,000 nodes and approximately 56,000 edges for the bladder cancer. And here I want to mention that those papers are not only coming from the medical domain, they are coming from computational pathology for other type of cancers as well. So why we selected to build a knowledge graph for the agent? Because we want to connect different ideas from different papers and different domains, and maybe benefit from some treatment which is given to some other cancer, to discover a biomarker for similar treatment for bladder cancer. So after we finish building the graph, now the path generation agent has the graph ready. So when we receive the human prompt, we take the prompt and we select the most important keywords in the prompt. For example, in this example, we have genes, we have tertiary lymphoid structure, and in the bladder cancer domain. So we take these keywords and try to find paths in the big knowledge graph. And you will end up with too many paths between those two keywords. So, but we need to select one of them. So the path generation agent select a random one. So if you take, there is a threshold as well, how random the path should be. So the shorter the path, it means the hypothesis will be very feasible, but it might not be novel enough. After the path generation agent, we have the ontologist, which takes the nodes and the connection between them and form the final hypothesis to give it to the scientist and then to the senior researcher and the human in the loop. So we'll talk more about the ontologist. So as I mentioned, you can select a random path, but there are too many paths. So a user might want to explore different hypotheses for the same input prompt. So we provide the option to generate multiple paths and then select the top end path. So there should be some criteria to select the best paths in this knowledge graph. So the candidate path can be selected based on its plausibility and the discovery. So we have two metrics for the plausibility, which are the logic and relevance, and we have two other metrics for the discovery, which are the novelty and the surprise. So for the plausibility, the logic, which measures the cosine similarity between the key nodes and connections on this path with the keywords we extracted from the prompt of the user. Then the relevance, which measures how close the nodes and the connections on this path to the central idea of the prompts, which is like the prognostic biomarker in bladder cancer in this case, the example. And the discovery, which has two metrics. The first one is the novelty. So the novelty metrics evaluate the rare nodes on the path. For example, if you have if you are asking for a relationship between genes and tertiary lymphoid structure, your different paths will have different genes. Some genes will be very popular, like TP53, for example, so it will be densely connected inside the graph, which gives it less novelty compared with another gene, which might be a rare gene and not well connected in the graph. The final metric, which is the surprise, this one measure the distribution of the path compared to the distribution of the full graph. For example, if the majority of the graph is genes and you have a path which contains all of the nodes are genes, this path will have less surprise compared with another path which connects concepts from the computational pathology like tertiary lymphoid structure or mitotic with the genes. So it has different domains, so the surprise will be higher. And finally, we aggregate all these scores in a single score to rank the different paths. So the weights are hyperparameter can be tuned by the researcher based on whether he wants to have more plausible hypothesis or more novel hypothesis. And we did some experiments to see whether the single path or multipath, like which one is better. So the multipath, it's very expensive. It will have like significant cost in terms of the total number of tokens because we are generating different hypotheses. And let's say we select the top 10 hypotheses. So we are running the path, the hypothesis generation whole pipeline for 10 times compared with a single random path. And the runtime will be more, of course. However, the novelty of the generated hypothesis will be higher than the one generated from a single path. And then now we'll move to the next agent. So we talked about the ontologist, we generated the path, the scientist took the path. generated the hypothesis. The hypothesis will get expanded by the senior researcher, then goes to the human approval. And if it's approved, it goes to the novelty critic. So the task basically of the novelty critic is to provide feedback about the novelty of the hypothesis. So we can't use only a single critic for scoring the novelty, because the critic might be very optimistic and overweight the novelty of the hypothesis, or the critic will be very conservative and give it very low novelty score. So we resolve this issue by designing a game theoretic adversarial debate between three different agents. So we have a prover who is like a very optimistic, designed to be an optimistic agent, which go to the research, pitch all the paper, and try to find gaps in research to prove that this proposed hypothesis is really novel. We have as well the verifier. So the verifier, it's the opposite. He is a conservative agent. It will go to the literature and try to fetch all the evidence which can disapprove with the prover, or find gaps in the proposed hypothesis itself. And we have the judge. So after each round, the judge take the input from both of the verifier and prover and try to come up with a decision. And after number of rounds, let's say 6 rounds, the judge will give the final answer. So those three agents, they depend on 6 scholarly databases to fetch the paper from, like PubMed's Semantic Scholar, Europe PMS, Bioarchive, and Archive, and cross-reference. So to make it clear, the first round of the novelty assessment start by asking each one of those agents to evaluate the novelty of the hypothesis separately, and they give their scores. So if the scores are almost similar, we don't go into the debate stage because it's expensive due to the token consumption and the running time. So we just give the average score as the score of the novelty, which is a number between 1 and 10. So we consider everything greater than 7 as novel hypothesis. But if the disagreement between them, or the standard deviation between the scores, is greater than 1, the debate will get triggered, and we'll start the debate between the prover, the verifier, and the judge. And we set up a maximum number of times for the debate. Here it's a 3 in our hypothesis generation pipeline. So if they reach agreement and the differences between them become less than 1, we converge and we give the final score. However, the verifier has the ability as well to flag this paper, this hypothesis, as might be suspicious hypothesis, and the judge upholds, and we apply penalty on this hypothesis. And the final score will be the average between the scores of the 3 different agents. So how did we test the debate-based novelty agent? So we depend on time travel backtesting to evaluate the hypothesis. So we collected around 150 proposals from literature, and we divide the data set into 5 tiers. So the first one contains papers which are foundational breakthrough, like AlphaFold 2. The second tier, which contains high innovation contributions. The third one, significant non-paradigmic work. And the tier D is incremental extensions of previous work. And the final one is the suspicious paper, like stab cell paper, which is proven later to be wrong. And during the time of debate, of course, we give the agents access to only the literature which happened before the date of publishing the specific paper. So we did the experiment where we used a single critic, and we found that the accuracy was 56 in classifying the paper properly. And the gaps between A, which is the breakthrough papers, and E, which is the suspicious one, was very small, 1.12, which means that this single agent was not able to distinguish between the breakthroughs versus suspicious papers. We used two critics where we used the verifier and the prover, but they don't have any debate between them, and we reach accuracy of around 75%, and even the gap increased between the A and E hypothesis. Then we use the multi-critic where we used three agents, but there is no debate between them. We just take the average score, and the accuracy even increased further, around 81%, and the gap increased as well. Finally, we took our debate agents, the full debate agent, and we reached around 91.3% accuracy, and the gap was the biggest among all the different critic agents. So, which shows the importance of the debate for giving the novelty of the hypothesis. So, is there any questions so far? Okay.
So I think there was a question on the chat, but it was more about it was about a few slides ago about the bladder cancer search with 400,000 papers. So I think the question was, I'm not sure if I got the number correct. Was it more than 100K for bladder cancer for the initial search, from which about 1,000 were selected?
Yeah, so going back to our graph, I think you are talking about this slide.
Exactly, yeah.
Yeah, so at the stage of the large-scale literature crawling, where we download all of these papers which might be related to that topic, the bladder cancer, we downloaded around, we found around 137,000 papers. But we didn't use all of them because we had other filtering steps to filter which paper might be related to the domain and which might not be related. So after all the filtering steps, we reached around 1,600 papers, which we used to build our graph.
Sure, thank you. It wasn't based just on PubMed. There was all these other sources as well.
Yeah.
Yeah. Okay, brilliant. I hope that answers your question, Constantino. Brilliant. Thank you.
Okay, great. Welcome. Okay, so the next stage is the validation stage. So after we give the hypothesis high novelty and it moves to the feasibility agent, it goes to the coding agent. So this might be very important contribution to for Sage, because other hypothesis generation algorithm might give you a hypothesis, but there is no way to check whether the hypothesis is really valid or not. So we added a coding agent, which consists of three different agents. The first one is the dataset discovery. So to be able to validate the hypothesis, we need a proper dataset which contains all the required files or images to validate the hypothesis on. So this part of the coding agent try to find if we have such data set in our repository, or if we can get it online and download the images and the related files. The second one is the execution step, where the coding agents try to generate the code for each of the subtasks, and it For example, if it needs some tools, it can access them from the tool orchestration subsystem. For example, to detect the tertiary lymphoid structure, you need already pre-trained tools, so you can access it from the set of tools we have. And then finally, we have after the execution, we get the results. So we need to interpret the results and understand whether the hypothesis is valid or not valid. So now we'll talk in details about each step. After getting the hypothesis from the feasibility agent, and we get a high score for that, we start with the dataset discovery agents. These agents will try to search for the proper format of the dataset in our repository, and then select all the related files or all the related dataset. And if it finds the proper dataset, then the task is over. But if not, it will ask the expert to interfere and might suggest a dataset or give a dataset to the agent. Then, after getting the dataset, we move into the code generation and execution step. So here, the LLM model will write the code, and it will, for some part of the steps, it will check whether we have the tool already available in our repository before coding it ourselves. Like, for example, in our hypothesis, the example we showed in the paper, we need a tool for the tertiary lymphoid structure, and we have it already in our orchestration subsystem. So this subsystem, it gets triggered by our coding agent. And here we have another LLM model and MCP orchestrator, which plans all the action should be taken by the agent. For example, if you want to detect the tertiary lymphoid structure on the whole slide image, first you need to call a batch extraction tool to perform to extract the batches from the whole slide image. Or you need as well the tumor detection tool, and you need the TLS detection tool. And when finishing, you return the tertiary lymphoid structure density values for all the patients in the dataset. Then the coding agent will move to the next step, which might be the survival analysis. So if the survival analysis tool is already available, we just run it on the data. If not, the coding agent will code the survival analysis part. So finally, the coding agent will return tables and plots. So we need to analyze them with another LLM to understand the plots and whether the hypothesis is valid or not. And finally, we validate our biomarker. So how to test the performance of our coding agent? So because there is no like a data set for hypothesis generation, we used a public data set called Karma Bench. So this data set contains 106 tasks to run on different domains, for example, coding mathematical tasks and other tasks as well. So this data set covers different type of problem. And what we found that we checked the execution rate for the coding agent and the exact match rate, which means how many times the coding agents get the right answer. And what we found that when we give the details about the dataset the agent should use, we have a high accuracy around 78.7 for the codex. But when we don't give the details about the dataset and the agent should do the data discovery part, the execution rate reduced significantly. Similarly for the exact match rate. So the data discovery part is very important for the coding agent to be able to perform all the other steps. And the failing in the code, we estimate the reasons for failing. So some of them might be due to the ability, the agent was not able to find the proper data set to perform the hypothesis. Some of them was that the agent found the data set but was not able to write a code to execute the task. Some of them were due to the execution error, and there was small number of them which has the exact right answer of the specific task. So after talking about all the different agents we have in the SAGE pipeline, I'll give an example on a valid hypothesis. So we tried to discover if we can find any biomarker which can relate specific genes and some image-based features to give a signature which is prognostic for overall survival in bladder cancer. So Sage came up with this hypothesis like high FAPP5 expression combined with scarce tertiary lymphoid structure aggregate predicts significantly worse overall survival in bladder cancer patients. So this hypothesis has a high novelty score. It was 8 out of 10, and it was feasible because the data set discovery agent was able to find a TCGA data set for bladder cancer, and it has around 412 patients. And we used all this data set in testing mode only. And as you can see from the results, the hypothesis was valid because the p-value was significant, and the hazard ratio was 2.34, and it doesn't cross 1, which means that the patient in the high FAPP5 and scarce tertiary lymphoid structure group had around 2.34 times elevated mortality risk. So you can see as well from the figure generated by the agent that the green one is the low FAPP5 and abundant TLS. Those are the low risk patients, and the high risk patients, the orange one, are the high FAPP5 and scarce TLS, and you can see the curves are like well separated, and the p-value is significant, and the hazard ratio is greater than 1. So that means after the agent which analyzed the results, we found that this is a valid hypothesis. So we compared as well our pipeline with different agents like ChatGPT for generating hypotheses, agent flow, which is built based on autogen, the same backbone for Sage, but here there is a planner and the agent will be like randomly called, but in our Sage we have a sequential order for performing the steps. So the validation showed that like Sage gives higher novelty, and the number of retrieved references for each generated hypothesis is lower, which means that the hypothesis is more novel, so it's very difficult to find resources similar or close to in the literature. We as well asked the urologist in our lab to score the hypothesis based on the novelty, plausibility, feasibility, implementability, and clinical impact and robustness. And the final score was between 1 and 5. And we instructed the urologist how to score each one of them. And it shows that for the urologist, he found that Sage was the best. So, of course, this was a blind evaluation. But we acknowledge that the number of generated hypotheses were small. So we are trying to validate our hypothesis generation on more number of generated hypotheses. So now I'll move to demos. So thanks to like the trainer who are working with me on this project, and Jason recorded this demo for the hypothesis generation pipeline, and later we'll see another demo on the orchestration subsystem. So if there is no questions on the pipeline, I'll just play the demo now.
Yeah, I think in the interest of time, we'll save our questions to the end.
Okay.
Cheers.
Hello, today I'm going to be demoing our hypothesis generation pipeline with an example prompt and the resulting output. The prompt we are going to be exploring is discover research ideas between tumor lysis syndrome and genes. First, our pipeline takes the input prompt and gives it to the path generation agent, which looks in our knowledge graph, in this instance, the bladder cancer knowledge graph, and looks for a specific path that links tumor lysis syndrome and genes. This information is passed to our ontologist, which defines the biomedical terms related for this path, and also the clinical relationships that are in the knowledge graph, which you can see here. And this information is passed to the scientist, which focuses on creating our main research proposal, including the hypothesis and other information. Here you can read the hypothesis for the prompt. And this information is passed to the hypothesis expansion agent, which takes the hypothesis from the scientist and comes up with a more expanded version, which is passed to our multi-critic coordinator, made up of 3 specialized critics with different personalities and search strategies to evaluate our hypothesis novelty using a debate-style framework. And this is in a sequential order, going from a prover to a verifier and the judge, and they debate to rigorously assess the outputs from different sources to calculate the novelty score. And the output for the multi-critic coordinator is quite long, so we don't include it here. And then the user decides whether to approve the current hypothesis or we can revise it. If approved, the hypothesis is sent to our explainability index agent, which scores the biomarkers used in the hypothesis based on different criteria and comes up with the overall score and an interpretation. Then the information is passed to our coding agent assistant, which creates a biomedical coding task in order to support computational validation of our hypotheses. And these are the 2 tasks that it generates. Once all of this is done, all of it is passed to our summary agent, which formulates a comprehensive review of all the aspects and critics after they have been expanded. And that is the overview of our hypothesis generation pipeline.
Okay.
Hello, today
Okay, so
hello.
Okay, so now the next demo, which is on the orchestration subsystem, so where the coding agent might prompt some of the tools existing in this system. So let's see the demo.
So this is a short demo of how to use this tool, the orchestration chatbot. So this is the dashboard that shows us the registered tools that are available for use. There are a few pipeline templates that are easily ready to execute. We also have the categories into which we have divided these tools into, and then we also have a viewer that can be used to view the whole slide images and also the output. So here's one test whole slide image that you can zoom into and that you can view, and then once you have the outputs, we can overlay them over this and we can view the outputs as well. This tools bar shows us all the tools that are available in this subsystem. So, and it also, once you click on that, you can also know more details about what this tool does and what the inputs are and what outputs would you get for every tool. And these are the pipelines that I spoke about, which are ready to execute. So you have a handcrafted pipeline, foundation spatial DL analysis pipeline that you can execute by clicking on it. And we can also create more using the chat that we'll come to here. So let's first try to run the patch extraction on the whole slide image that we just looked at. We need to give the input directory, and then it'll give us the results, and it'll store it in the output directory. So we'll run the patch extraction on the whole slide image that's stored at. We'll just give. Now this runs in the backend on the V100 GPU for now, and then we'll get this following result. So here, as you can see, we have successfully gotten the output for the patch extraction. It gives some details about how many coordinates and what were the tumor, tissue, periphery patches, and it also says where it saves the outputs. And then even once you go to the viewer, we should be able to click on this. And then once we click on the overlay, we can click on the coordinates, and then we can see that the red ones are the tumor, the yellows are the periphery, and the green ones are the tissue for this particular whole slide demo image that we took. So this.
Okay. So now we come to the limitations, the contributions, and the takeaway of this talk. So we acknowledge that our work has many limitations. So the dataset discovery part, for example, is very important for the coding agent. And we notice that when we don't give some information about the dataset, the performance will drop for the different coding agents from around like 24 to 44%. So we need to spend more time to optimize the dataset discovery agent. Another limitation is the generalizability of our pipeline. So far, we tested our pipeline on bladder cancer only, but we need to show its generalizability to other domains as well. The evaluation samples of like some of the experiments we did as the comparison between different hypothesis generation method was a small number, so we need to evaluate it on a bigger number of hypotheses. So to summarize our contribution, we created SAGE and we empowered it with the knowledge graph. So for bladder cancer, we designed a knowledge graph from 1,650 papers. And we proposed the multi-path ontologist, and we allow the user to select not only a single path from a single prompt, but many of them, and we rank them as well based on the plausibility and the discovery aspects. We have as well proposed the debate agents, and we showed that it significantly improved the judgment of the novelty of the hypothesis compared with a single agent or the agents which work on each own, like without interacting with each other. And finally, we have the main contribution, which is the coding agent, to be able to run the hypothesis and validate whether the hypothesis is valid or not on a large data set. Like, for example, in our example on TCGA data set, we evaluated on more than 412 patients. The takeaway that SAGE is a closed-loop agentic framework for interpretable biomarker discovery, and it will speed up the biomarker discovery and advance the domain for the biomarkers in the medical domain. So finally, I'll end up the talk with thanking all the members who worked with us on SAGE and the members as well who are working on Agentic AI now, and they helped with preparing the demos. So thank you, everyone. Any question?
Well, thank you very much, Sal, for a really, really interesting talk there.
Thank
We haven't got much time left, but we've got time for a couple of questions, I think, still. But no, that was a really, really interesting talk. So thank you so much for giving your time there. So if there's no questions on the chat or in in the room, Jachi, then I'll start by asking the first question. So, of course, in your pipeline there for Sage, you've got a whole host of agents, right? I'm
you.
just wondering if you'd managed to sort of, and obviously you validated each of these individual agents or sort of groups of agents individually, and then you've obviously at the end you've got this complete pipeline that you've sort of gone from A to B, and you've shown that it works. I'm just wondering if you could potentially give us any more details about how you, well, I'm just wondering if you could, whether you tested taking out some of these agents, whether some of them are overkill and you don't necessarily need quite as many, because you've
Yeah.
got 10, 15, 20
Yeah.
different
So
agents at parts, or
yeah, exactly. So we don't show the ablation study here, but we started with many agents for the initial pipeline, and we noticed that the hallucination increases so much. So it might give you a hypothesis, and it suggests what should be the, for example, the hazard ratio, and give you lots of details about p-value, etc. So, which considered as the hallucinations because of those different agents. So, what we did, we tried to prune the number of agents to get to the smallest number. Like instead of getting like three senior researcher, we just replaced them with one. Besides, the input from for each agent is very important. So previously we were giving one agent input from all other agents, and it turned out it confuses the agent and consumes so much tokens. So by just doing sequential like prompting of the agents, we reduced the cost by 75% for the token consumption. So yeah.
oh, brilliant. And I wasn't sure if I missed, was it that, so the individual agents that you're using, are they ChatGPT, did you say, or was it you're using AutoGen? I don't know if I missed that.
Yeah, so the backbone for all the agents is the autogen, but you
Okay.
have, yeah, the ability to select what agent you want to use. So you can use agents like API keys to prompt ChatGPT, or you can use Claude or Gemini. So you have the ability to select which one you want to use.
Okay, brilliant. And the final sort of agent, I think it was a summary agent, but in reality, can it also do you think you could add an agent to write the paper at the end as well? So you've got a full pipeline there from posing a question to having experiments and a paper written.
Yeah, sure. So this is the aim, like to make it automatic, like fully automatic without a human interaction. The human interaction only is for like the medical domain is very sensitive, so we want like human feedback, and the feedback will be used in the future maybe to improve the performance of the hypothesis generation. So yeah, but we found as well if you include agents from different type of agent like ChatGPT, Gemini, and Claude with each other, it will perform better than having three ChatGPT agents. So yeah.
No, brilliant. And so in your lab now, then, do you have now this pipeline's working well? Is it pumping out lots of papers then?
Yeah, hopefully, yeah.
Okay, brilliant. Has anyone got any questions in the room, Jachi, or anyone else online?
Yeah, okay. I have a quick question about the knowledge graph. So have you tried different ways of constructing the knowledge graph? And with this graph, does it have to be updated like every now and then? And how often do you think it has to be updated? And how long will it take to update this graph to keep up with the recent research?
Yeah, so building a knowledge graph, like the time can, it can, like, it might take 1 week based on the resources we have and the availability of these resources and the number of papers as well. So you're right that adaptation of knowledge graph, adding more papers is very important. So we haven't done that yet. So we are planning to do that and merge as well knowledge graphs from different domains. so that we can maybe do like cross-cancer biomarker discovery. Yeah, but we haven't like done many experiments on the different ways to construct the knowledge graph. Yeah, I'd be happy to hear your thoughts if you have any suggestions for this.
Yeah, I don't really have any suggestions. Yeah,
Yeah, it's nice
a,
experiments for the future. Yeah, thank you.
thank
Brilliant. I suppose one final question then, just to finish us off then, Sahar. So we're obviously seeing agentic AI sort of absolutely explode at the moment, right? And there's loads and loads of new papers coming out. What do you think are the biggest challenges over the next 12 months for sort of research in agentic AI, especially in computational pathology and medical imaging?
Yeah, so like creating the agents might not be that difficult and combining them, but like the question you are asking and the design of the experiments is very important. So it needs domain experts to do that. So this is very important. And the agents, as you saw for the coding agents, still have lots of limitations. So the dataset discovery is very like a bottleneck in this coding agent part. So people need to work more on it and maybe think of ways to provide agents with memory so we can reduce the time of generating hypotheses. Yeah, and reinforcement learning is a very hot topic now for teaching agents from experience. Yeah.
Sure. Okay, then brilliant. Well, I think with that, we should probably draw the seminar to a close. But thanks again, Sal, for a really, really interesting talk there.
Thank you.
Yeah, and a bit of a reminder for everyone online. So our next seminar is scheduled for the Monday, 27th of April, and we have a talk by Dr. Juan Zian from Fudan University. So hope to see you all there. But again, thank you very much, Sahara, and thank you everyone for joining us online and in person.
Thank you. Yeah, bye.
Bye, everyone.
Bye.